Imagine just staring down a microscope at this tiny slice of lung tissue, knowing that your completely subjective guess about like a chaotic pattern of cells is going to dictate whether a patient receives aggressive chemotherapy or if they just go home.
Yeah, that is a huge amount of pressure.
Right. But that is literally the nightmare that human pathologists face every single day with this specific, highly aggressive lung cancer called invasive pulmonary adenocarcinoma, or IPA. So welcome to today's deep dive. I'm really glad you're here with us because today we are looking at how artificial intelligence is, well, finally solving this.
And it really is a massive breakthrough.
It really is. We've got this super dense stack of research today. It's a groundbreaking multi-center study that was published in partnership with the Hormel Institute, which is part of the Nature portfolio. And our mission for you today in this deep dive is to unpack this bio-interpretable ensemble learning model. Basically, an AI that grades this cancer using macroimaging, microimaging, and genomics all at once.
Yeah, and what's so fascinating here, I mean, to me at least, is that this research isn't just about, building another AI that guesses a diagnosis. It's really about cracking the black box of artificial intelligence in medicine.
Which has always been the huge complaint, right? The black box.
Exactly. For years, algorithms have been making these predictions, but doctors couldn't actually see how they arrived at those conclusions. But this study, it bridges that gap by physically linking the pixels in an image to the invisible genetic mutations that are actually driving the tumor.
Okay, let's unpack this, because before we get into how the algorithms are actually solving the problem, we really need to exhaustively understand the clinical bottleneck that human doctors are stuck in right now.
We have to understand the problem before the solution.
Right, so let's ground this in a real-world scenario from the data. Say a patient undergoes a radical surgical resection. The surgeon removes this lung tumor, right, and sends it to the pathology lab. The pathologist gets a stained slide of this tissue. They sit down at their microscope, and what mechanically are they looking for? Why is it so incredibly difficult?
Well, to really grasp the difficulty, we have to look at how IPA actually behaves. So IPA is not this uniform mass.
Okay.
It's highly heterogeneous, meaning it's just this chaotic, tangled mix of different cellular architectures that are all growing at the exact same time. And to make sense of this, pathologists use a grading system developed by the IASLC.
That's the International Association for the Study of Lung Cancer.
Exactly. Yeah. And they categorize the tumor's appearance into 3 distinct grades. And this is based entirely on the physical growth patterns of those cells.
And the distinction between those grades, that's critical for the patient's survival.
Oh, absolutely. It dictates the entire treatment plan. So grade 3 is called the lipidic pattern. Think of cancer cells growing neatly along the existing alveolar walls of the lung.
Like scales on a butterfly wing or something?
That's a perfect way to picture it, yeah. It's relatively slow moving. But then grade 2 steps up the aggression. This includes acinar and papillary patterns where the cells start mutating and they form these little glandular structures or fern-like fronds.
So it's getting messier.
Much messier. And then grade 3 is the true danger zone. This includes solid patterns, which are literally just dense bricks of tumor cells, and the micropapillary pattern.
Micropapillary, what does that look like?
That's where the cells form these tiny floating tufts that don't even have a central blood vessel. Finding grade 3 patterns means there was a really high chance of recurrence and a very poor prognosis. It basically triggers the immediate need for heavy chemotherapy.
So the pathologist is just hunting for these patterns visually, which sounds tough, but they are highly trained experts, so why exactly is this considered a clinical nightmare?
The bottleneck really comes down to what we can call the 5% rule.
The 5% rule.
Yeah, so about 80% of all IPA tumors don't just have one of these patterns. They have 2 or more mixed together. So the pathologist's job isn't just to look at it and say, oh, I see micropapillary cells.
Right.
They are actually required to visually estimate the exact percentage of every single pattern present in the entire tumor in 5% increments.
Wait, I want to make sure I'm visualizing this correctly. You're telling me they have to look at a slide that has millions of overlapping cells and manually just calculate in their head, this looks like 15% micropapillary, 45% acinar, and 40% lipidic.
That is the exact clinical expectation, yes.
That's insane.
It is. And the human visual system is simply not built to calculate percentage breakdowns of chaotic overlapping textures objectively. And the study provides some pretty stark evidence of this using kappa scores.
And kappa scores, just to clarify for everyone listening, that measures inter-observer agreement, right? Like, how often two expert pathologists look at the exact same slide and give the exact same grade?
Correct. With the score of 1.0 being absolutely perfect agreement. Now, for the solid subtype, those dense bricks of cells we talked about, the agreement is substantial. It hits a 0.756.
Okay, because it's a giant block of cells. It's easy to see.
Right. But for the micropapillary subtype, which is the most dangerous one, the score just plummets to 0.143.
0.143.
Yeah. That is officially categorized in the literature as poor.
A 0.143 agreement rate on the deadliest cancer pattern. That is terrifying. It really reminds me of, those carnival games where you have a massive glass jar just filled with jelly beans?
Oh, yeah.
But in this case, the jelly beans are all slightly different shades of pink and red. And we're basically asking a human to just eyeball this massive jar and confidently state exactly what percentage are cherry, what percentage are strawberry, and watermelon, down to a 5% margin of error. It is completely guaranteed to be subjective.
It is an impossible ask. And it's actually further complicated by physical artifacts.
What do you mean?
Well, when a biopsy is taken, lung tissue often just collapses. The pressure of the biopsy literally changes the shape of the cells on the slide. So a harmless lipidic pattern might suddenly look crowded and dense, perfectly mimicking a dangerous solid pattern.
Oh, wow.
Yeah, that morphological overlap makes human consistency virtually unattainable.
It's wild to step back and look at the gravity of this. I mean, for you listening right now, just think about how much weight we put on a medical diagnosis. So let's do a quick recap of where we are. Bullet 1, IPA grading relies on estimating these tiny percentages of totally mixed cell patterns. Bullet 2, human pathologists really struggle with the consistency here, especially with those deadly micropapillary patterns hitting that 0.143 kappa score. So my question to you, the listener, is have you ever considered just how subjective medical grading can actually be, even among the most seasoned experts?
It really highlights a systemic vulnerability in pathology as a whole. Because if human vision is inherently subjective when dealing with this level of chaos, the logical next step is we have to remove human vision from the equation.
Right, teach a machine to see it.
Exactly. We need to teach a machine to see the tumor objectively. But a tumor exists on multiple physical scales. We can't just look at it in one way.
Which brings us to the whole multi-omics approach of this study, combining radiomics and pathomics. We are essentially attacking the visual data from two completely different altitudes. Let's start with the macro view, radiomics.
So radiomics looks at the entire forest. Before a patient even goes into surgery, they get a CT scan of their lungs. The researchers here use this open source software called PyRadiomics to extract what they call handcrafted features from these scans.
Handcrafted meaning what exactly?
When we say handcrafted, we mean mathematicians and doctors literally sat down and wrote specific equations to look for known biological markers. For instance, they wrote an algorithm searching for a feature called small area, high gray level emphasis.
Okay, so breaking that down, it's mathematically hunting for small pockets on the CT scan that are extremely dense and opaque because cancer tissue is physically denser than healthy lung tissue, right?
Correct. Yeah. They also wrote algorithms to find the minimum pixel intensity, which helps define those hazy sort of ground glass borders of the tumor. But human math can only describe so much. To capture the rest of the data, they ran the CT scans through a deep learning neural network called ResNet-18.
ResNet-18?
Yeah. This AI isn't given any rules. It just scans the pixels and automatically extracts these hidden, abstract textures that human language doesn't even have words for.
Okay, so that gives us the macro environment of the tumor in the lung. Now we need to zoom all the way in to the micro level, which is pathomics. This is looking at the individual trees using those whole slide images of the stained tissue we talked about earlier.
And again, they use a two-pronged approach here. First, handcrafted features using software called Cell Profiler.
I've heard of Cell Profiler.
It's incredible software. It identifies individual cell nuclei on the slide and calculates things like Zernike-shaped features.
Zernike-shaped features, what is that measuring?
So Zernike polynomials measure the exact mathematical symmetry or asymmetry of a cell's nucleus. A healthy cell is relatively uniform, right? But a mutating cancer cell becomes deformed. They also measure texture entropy, which quantifies exactly how chaotic the pixel distribution is inside the cell walls.
So we have the handcrafted microfeatures, but the deep learning side of pathomics, this requires some serious unpacking. Because a whole slide image, a WSI, that is basically a gigapixel image, isn't it?
It is.
It contains billions of pixels. You can't just upload a gigapixel image into a standard neural network. The computer would just instantly crash.
Oh, they would immediately run out of memory.
Yeah.
And this is where a paradigm called multiple instance learning, or MIL, becomes incredibly crucial. Think of a whole slide image like a massive, highly detailed satellite map of an entire country.
Okay.
The AI can't process the whole country at once. So MIL breaks that massive image down into thousands of tiny manageable tiles or patches. In this study, each patch was exactly 1024 by 1024 pixels.
So we literally slice the slide up into thousands of microscopic squares. Then what happens?
Then the neural network looks at each individual patch and assigns it a probability score. Basically asking, is this specific patch showing high-grade or low-grade cancer?
Okay, but a patient doesn't get a diagnosis per patch. They need one single diagnosis for the whole tumor.
Exactly. So the MIL algorithm aggregates the probabilities of all those thousands of patches into one final score. And the genius of this system is its attention mechanism.
How does that work?
It mathematically assigns a higher weight to the most aggressive patches. So if 90% of the slide is low grade, but the AI finds a few patches of highly aggressive grade 3 cells, it forces the final diagnosis upward, because those aggressive cells dictate the patient's actual survival risk.
I have to play devil's advocate for a second here, though.
Sure.
We have handcrafted features, which are essentially rigid human equations based on known geometry. And we have deep learning features, which are entirely abstract patterns the AI just finds on its own. Aren't these two systems fundamentally incompatible? Like, it feels like mashing a highly structured math textbook together with an abstract painting.
It might seem that way on the surface, but they're perfectly complementary.
Really?
Yeah. If you only use deep learning, you get a very powerful model, but it's brittle. If the hospital uses a slightly different CT scanner, the abstract patterns might change and the AI just fails.
Oh, I see.
But handcrafted features like cell shape or tissue density, those are grounded in physical biology. They don't change based on the camera you use. So by fusing them together, you anchor the raw pattern recognition power of deep learning with the physical stability of human knowledge.
So to gather our thoughts here for a second, let's look at what we've covered. We are tackling this massive subjectivity problem by hitting it from absolutely every angle. So bullet 1, radiomics looks at the macro CT scan, it's the forest. Bullet 2, pathomics looks at the microcellular slide, the trees. And bullet 3, MIL algorithms stitch thousands of tiny microscopic patches into one cohesive diagnosis. It really puts the complexity of this disease into perspective. So if you were designing this AI, listener, which level of data, the macro CT or the microslide, do you think holds the most crucial secret to the tumor's behavior?
Well, if we connect this to the bigger picture, the answer is that neither is actually sufficient alone. The secret is in how they interact with each other.
Right.
But right now, at this stage in our process, we have a massive data problem.
Yeah.
The researchers extracted thousands of different mathematical features from these images, and most of them are just noise. So how do you filter out the noise to build a functional tool?
Right, because you can't feed thousands of overlapping noisy variables into a diagnostic model. It would just confuse the system. So how do they narrow it down?
Through an exhaustive, multi-stage mathematical gauntlet.
A gauntlet. I love that.
First, they used ANOVA, that's analysis of variance. They used it to immediately throw out any features that had no statistical difference between high-grade and low-grade tumors. I mean, if a feature doesn't change when the cancer gets worse, it's useless.
Makes sense.
Then they deployed the Bruta algorithm.
The Bruta algorithm is fascinating to me. It's a random forest wrapper, but the way it works, it's almost like a stress test for the data itself, right?
Exactly. It creates what are called shadow features.
Shadow features?
Yeah. It takes a real feature, copies it, and then randomly shuffles the data to create a fake shadow version of it. Then it pits the real feature against the shadow feature.
Wow.
If the real feature can't predict the cancer better than the randomly shuffled fake data, the Bruta algorithm just throws the real feature in the trash.
That's amazing.
It ensures that every remaining variable is genuinely impactful, not just some statistical fluke.
And then the final boss of this gauntlet is Lasso.
Yes, Lasso, the least absolute shrinkage and selection operator. Lasso is brilliant because it actively penalizes a model for being too complicated.
Oh, so?
Well, if two features are basically telling the AI the exact same thing, Lasso shrinks the mathematical coefficient of the less important feature all the way down to absolute zero, just removing it entirely.
So it forces efficiency.
Exactly. It prevents a model from overfitting, which is when an AI basically just memorizes the training data but completely fails in the real world.
It's just a ruthless elimination process. We started with thousands of data points. After ANOVA, BRUTA, and LASSO, what actually survived the gauntlet?
Exactly. 35 optimal features.
Right.
That is the golden data set. So 11 deep learning radiomics, 5 handcrafted radiomics, 5 handcrafted pathomics, and 14 deep learning pathomics.
And these 35 elite features become the foundation for the final predictive model, the DHRP's model, which stands for Deep Learning and Handcrafted Radiopathomics.
Exactly. And to build this model, the researchers didn't just pick one AI framework. They tested 11 different machine learning algorithms using these 35 features. They tested support vector machines, random forests, XGBoost, and a multi-layer perceptron, or MLP.
And how do they do?
The MLP actually performed the best on its own. But this raises a really important question. If you are trying to make a foolproof medical decision, why rely on one single algorithm?
You don't. You use ensemble learning.
Yeah.
This is honestly one of my favorite concepts in computer science because it's the exact same logic as assembling a medical board. Imagine you have one genius doctor diagnosing a patient. That doctor might be incredibly smart, but they might have a personal bias, or, they might just be tired that day. But if you put 11 super specialists in a room, give them all the exact same data, and have them cast a vote, the consensus of that group is going to be far more accurate and stable than any single individual.
The DHRP's model operates exactly like that medical board.
Yeah.
It combines all 11 machine learning algorithms into a weighted voting system, so it eliminates the algorithmic bias of any single framework.
Yeah.
And the performance of this voting committee was just staggering.
Let's talk numbers.
So we measure this using AUC, or area under the curve. That plots the true positive rate, the correct diagnoses, against the false positive rate, which are the false alarms. A perfect score is 1.0. In the training set, the DHRP's model hit an AUC of 0.982.
That's incredibly high. But training data is one thing. How did it perform on unseen data from completely different hospitals?
In the independent testing sets, it maintained a highly robust AUC between 0.885 and 0.920. But the ultimate test was putting this AI head-to-head against human expertise. They had 2 board-certified pathologists with over 15 years of experience review the exact same cases.
Who won?
The AI matched or beat them. Both veteran pathologists exhibited lower sensitivity than the AI, meaning they actually missed more of the true high-grade cancer cases than the algorithm did.
Wow.
Furthermore, the model achieved significantly higher specificity than one of the pathologists, meaning it was much better at correctly ruling out false alarms.
Okay, so let's synthesize this for a second. We have taken a subjective, error-prone 5% guessing game. We filtered the visual noise through ruthlessly efficient algorithms like BRUTA and LASSO down to just 35 features. And we fed them into an 11-algorithm voting committee that is actively outperforming doctors with decades of experience.
Total paradigm shift.
It fundamentally shifts how we view diagnosis. So, bullet 1, algorithms filtered the noise to 35 key features. Bullet 2, the DHRP's ensemble uses 11 algorithms voting together. And bullet 3, the AI matched or outperformed veteran human pathologists. Think about it, listener. If you were sitting in that clinic waiting for a life-altering cancer grade, would you feel more secure trusting the visual estimate of one human doctor or the calculated consensus of an 11-algorithm AI ensemble?
It really is a profound shift in trust.
Yeah.
But there's a massive roadblock here. The medical community inherently distrusts black boxes.
Right.
It is not enough for an AI to just be highly accurate. We have to know why it is making its choices. What is the actual biological reality behind the 35 pixels and shapes it chose?
And that leads us to the final and, frankly, I think the most mind-blowing piece of this entire study, transcriptomics or RNA sequencing. This is how they proved the AI wasn't just doing mathematical parlor tricks, right?
This is the crux of the study's brilliance. They didn't stop at the images. They took the actual tumor tissue from the patients in the study and sequenced their RNA to see exactly which genes were turned on or turned off.
But you can't just look at thousands of individual genes. That's too chaotic.
Way too chaotic. So they used a technique called WGCNA, Weighted Gene Co-Expression Network Analysis.
Let's break down WGCNA because this is vital. It's a form of clustering, right?
Yes, because biology doesn't operate in a vacuum. Genes don't work alone. They work together in these intricate networks. Imagine trying to understand how a massive factory operates. You wouldn't evaluate the factory by just watching one random worker on the assembly line, right?
Right. You'd look at how whole departments communicate and work together.
Exactly. WGCNA takes thousands of genes and mathematically clusters them into modules based on how they co-express, meaning which genes turn on and off at the exact same time. The researchers identified 11 different modules, but 2 of them, which they color-coded as the green module containing 273 genes and the yellow module containing 309 genes, those 2 correlated perfectly with the IPA grading.
Okay, so if the green and yellow departments, so to speak, are driving the cancer's grade, what exactly are those 500-plus genes actually doing?
Well, in high-grade aggressive IPA, the genes regulating the cell cycle, the pathways responsible for cell proliferation and division, were universally upregulated in these modules. The cancer cells are literally keeping their foot on the gas pedal, multiplying out of control.
Just nonstop division.
Exactly. And conversely, the genes responsible for transmembrane transport and endosomal functions were significantly downregulated.
Wait, what does downregulating transmembrane transport mean for the tumor physically? Like, what does that do?
It means the tumor cells are fundamentally altering how they interact with their environment. By shutting down normal transport functions, the cells enhance their migratory abilities. They detach more easily. They're gearing up to invade surrounding tissue and metastasize.
Here is where it gets really interesting. Because the researchers took those 35 image features the AI selected and mathematically mapped them to these specific gene modules.
Yes.
And they found that the physical shapes the AI was looking at on the screen directly linked to 6 specific biological pathways, including signal transduction, cell proliferation, and metabolism.
Yes. Let's look at the CT scans first. The macroscopic features, those dense patches the AI found on the CT, they link heavily to signal transduction, specifically the aberrant activation of the MAPK/ERK pathway.
The MAPK/ERK pathway.
Yes. When this genetic pathway mutates, it causes the cancer cells to form these dense clusters and triggers a fibrotic scarring reaction in the surrounding healthy lung tissue.
And that fibrotic scarring physically manifests on the CT scan as spiculation, right? Like those spiky, dense edges on the outside of the tumor.
Exactly.
So when the AI flags a spiky border on a CT scan, it isn't just looking at geometry. It is literally seeing the physical footprint of the MAPK/ERK genetic mutation.
That is the exact breakthrough. And it happens on the microscale too. The pathomics features from the whole slide images linked heavily to metabolism. When a tumor becomes highly aggressive, it undergoes metabolic reprogramming to fuel its rapid growth.
Okay.
This massive shift in energy consumption physically alters the architecture, the shape, and the internal texture of the cell itself.
Which is exactly what the Zernike shape features and texture entropy algorithms were picking up. The AI is inadvertently diagnosing invisible genetic mutations and metabolic states purely from a photograph.
It is decoding the internal molecular mechanisms through external morphological phenotypes. It completely demystifies the black box. The AI isn't guessing. It is recognizing the physical destruction caused by specific genetic warfare.
It is unbelievable. We've gone from human doctors struggling to estimate mixed patterns to an AI that can look at a spiky border on a CT scan and just say, hey, that tumor has a MAPK/ERK pathway mutation, and its metabolism is actively reprogramming to metastasize. It completely changes your perspective on what an algorithm is actually seeing.
If we synthesize all of this, what we are looking at is a roadmap for the future of personalized medicine. Multi-scale, multi-omics models like this DHRP's ensemble, they provide an objective, reproducible benchmark.
Yeah.
We eliminate the subjective human guessing game, but much more importantly, we provide doctors with biological interpretability. We finally understand the why.
Now, to be thorough, what are the caveats of this research? Because we always need to ground these massive leaps in reality. There are always limitations.
Oh, definitely. And the study clearly acknowledges its limitations. The most critical one is a lack of spatial resolution in the genomics.
What does that mean exactly?
They used bulk tissue profile. That means they took a piece of the tumor and essentially ground it up in a blender to sequence the RNA. It tells you what genes are active, but it completely destroys the map of where exactly in the tumor microenvironment those genes were firing.
Right, because it's all blended together.
Exactly.
Yeah.
To truly perfect this biological link, future studies need single-cell sequencing or spatial transcriptomics, which preserves the cellular zip codes, so to speak. Additionally, while this was a multi-center study, AI models always need larger, more diverse external validation sets across different global hospital systems.
Oh, to make sure it generalizes.
Right, to ensure the algorithm doesn't get confused by different brands of CT machines or different chemical staining protocols in the lab.
So to recap this final section, Bullet 1, WGCNA clusters genes into actionable modules like green and yellow to understand tumor behavior. Bullet 2, microscopic CT features directly reflect internal signaling pathways like MAPK, ERK. And bullet 3, microscopic WSI features actually decode the tumor's metabolic reprogramming. So how does it change your perspective on AI, knowing it can literally see invisible genetic pathways just by looking at the physical texture of a standard CT scan? We started this deep dive exploring a massive vulnerability in medicine, the sheer subjectivity of human vision when faced with microscopic chaos. And we've seen how layering radiomics, pathomics, and genomics can solve it. We have a committee of algorithms filtering out the noise, beating human experts, and proving their work by linking pixels on a screen to the very DNA of the cancer.
It transforms pathology from an observational art into a quantitative, biologically grounded science. It ensures that a patient's treatment plan is based on objective data, not optical illusions.
Which leaves us with a final thought to ponder as you go about your day. If a machine learning algorithm can look at the physical architecture of a tumor on a screen and accurately deduce the chaotic, invisible genetic warfare happening inside its cells, what other hidden biological truths are currently sitting in plain sight just waiting for the right algorithm to decode them?